Alright, good morning boys. So far we have discussed about change of chromosome number and structure. In that we have discussed about polyploidy, in which the entire set is more than twice. It could be more than 3, more than 2, it could be 3, it could be 4, it could be 5, it could be 6. Then we have talked about aneuploidy. Aneuploidy is change in the chromosome number, that is addition or deletion of one or more chromosome. Not the set, but one or more chromosome. In that we have discussed about hyperpolyploidy and hypopolyploidy. Now we will talk about the change of structure of chromosome. This is what we have left in the last class, change structure of chromosome. When we talk about structure of chromosome, it is the chromosome that is there. It has a particular sequence. Genes are arranged on the chromosome in a particular sequence. When there is a change in the sequence of the chromosome, that is what is called change of structure of chromosome. It is also called chromosomal aberrations. It is also called the chromosomal aberration. Now, when we talk about chromosomal aberrations, we talk about change in the sequence of the genes on the chromosome. It could be change of the structure of chromosomes. This is also called chromosomal aberrations. This is called chromosomal aberrations. Change in the structure of the chromosome. That is, change in the sequence of the chromosome. Sequence of the genes on the chromosome. In that, we'll talk about the first one that we're going to talk about is duplication. Duplication. Take for example, you have a chromosome with the centromere here. Let's say the genes are A, B, C, D, E, F, G, H. This is the sequence of the chromosome. This is the normal sequence of the chromosome. After duplication, let's say the chromosome, the gene AB is duplicated. Then after duplication, the sequence looks like this. Obviously it will be slightly longer. It will be slightly longer. This is the centromere again. We'll have AB, AB, C, D, E, F, G, H. So what you see here is this particular part of chromosome is duplicated. That means there is an extra gene of AB that is present. This is called duplication. The other case is called, the second case is called deletion or deficiency. Deletion or deficiency. Deletion or deficiency. Now we take the same, let's take the same gene sequence A, B, C, D, E, F, G, H, and after deletion, what happens? Let's say again AB is removed. This is called deletion. Whenever a small portion of the gene is removed from the chromosome, that is called deletion. Next we have is the translocation. Translocation takes place between two non-homologous chromosomes. Two non-homologous chromosomes. Let's say this is the first chromosome, the non-homologous chromosome, the other is non-homologous chromosome, as we know, are of different shape and sizes, and the genes are also different. So let's take for example I, J, K, L, M, N. This is the non-homologous chromosome. This is the non-homologous chromosome. Now what happens here is there is a segment that is deleted from one of the chromosome and added to the non-homologous chromosome. Let's take for example this portion. This portion is deleted and now it is added here. AB comes here. This is called translocation. Sorry, this is called deletion. We have finished. This is called the third type, translocation. This is called translocation. That means this part is removed from the chromosome, and that is, it goes and sticks with the non-homologous chromosome. This is called translocation. Now the last of the change in the structure of the chromosome we talk about is the inversion. In inversion, there is a 180-degree rotation of the gene. In the same chromosome, there is 180-degree rotation of the gene. Take for example, again to make things simple, we'll stick with ABCD. This is the centromere. A, B, C, D, E, F, G, H. This is the sequence of the gene on the chromosome. Now in inversion, what happens? There is 180-degree rotation. This particular segment, let's say, after inversion, it looks like this. A, B, again I have the centromere here. A, B. Now the F will come here because it is 180-degree rotation. F, E, then what goes here is D, C, G, H. So what you see here is a 180-degree rotation. This is what is called inversion. These are the four different types of change in the chromosomal structure of the chromosome, which is also called chromosomal aberration. Now in this segment we are going to talk about, in brief, about prokaryotic chromosome and eukaryotic chromosome. We are going to talk in brief because we already know what is a prokaryotic cell and an eukaryotic cell. This we have talked about in the class 11 syllabus. Now we'll talk about in brief about what is the difference between prokaryotic chromosome and eukaryotic chromosome. Now the prokaryotic chromosome we already know is a primitive type of chromosome, or we can say the first form type of chromosome, is double helical, it could be double helical or circular DNA or circular DNA or double helical or circular DNA not enclosed by a membrane. Not enclosed by a membrane. We already know in a prokaryotic cell, the nuclear membrane is absent, so the nuclear material remains suspended in the cytoplasm. So there is no membrane. That means bacterial cell is always in a haploid state. Bacterial cell, we find this in a bacterial cell which is always in a state of haploid. Bacterial cell is in a state of haploid. Obviously, there is no protein. That means the histone proteins which are normally present in the eukaryotic chromosome is absent in the prokaryotic chromosomes. Another important feature that we see here is no introns. Now normally in a eukaryotic chromosome we have introns and exons. The exons are the genetically active part whereas introns are just an intervening sequence which is made up of repetitive DNAs. This introns is absent in the case of bacterial chromosome or the prokaryotic chromosome. This no introns, that means introns are basically non-coding DNA sequence, non-coding DNA sequence which is absent in the case of prokaryotic chromosome. Now, another important feature is the rate of division. The rate of division is very high. No wonder the organisms like bacteria having prokaryotic chromosome can multiply very quickly because their rate of division is very high. Now we go on to this is in brief about the prokaryotic chromosome. Now we go into brief about eukaryotic chromosome. Eukaryotic chromosome, as we know, are rod-shaped or thread-like with nuclear membrane. That means eukaryotic chromosome are rod-shaped or thread-like and covered by nuclear membrane. This we already know. In the case of eukaryotic cell, we know a clear, well-defined nuclear membrane is present. That means the nucleus is present. The nucleus with a nuclear membrane is present. Whereas in the prokaryotic chromosome, there is no nuclear membrane. So in that case, the nucleus is called the nucleoid. Another important feature here that we see is presence of centromere. In an eukaryotic chromosome, we have the centromere present, to which the arms are joined together. To which the arms are, or we can say the centromere is present at the center of the arms, and it normally has one or two arms. Another important feature that we see here is the chromosome is made up of DNA and histone proteins. Histone proteins, which forms the core in which the DNA is bound. That is why they have nucleosome as well. Nucleosome is also present in the case of eukaryotic chromosome. There is also a nucleosome which is present. They have histone proteins as well. Remains invisible. An eukaryotic chromosome remains invisible during the interphase. That means we can see different cycles of the chromosome in the different stages. It remains invisible in the interphase. It becomes visible in the metaphase stage. It condenses and becomes visible in the metaphase stage. It has both introns and exons. It has both introns and axons. It has both, they are invisible during interphase and become visible during metaphase. They become visible during the metaphase. They have introns and axons present. As we have already talked about, introns are the intervening repetitive DNA sequences, whereas axons are the main, you can say, coding sequence which are genetically active, which helps in the synthesis of proteins. and they are made up of complex nucleoproteins called the chromatin, composed of euchromatin and heterochromatin. They are made up of complex nucleoprotein called chromatin, and this chromatin is of two types, euchromatin and heterochromatin. Chromatin and heterochromatin. Euchromatin and heterochromatin. After the prokaryotic and eukaryotic chromosomes, let's talk about the euchromatin and heterochromatin, which is present normally in the case of eukaryotic chromosome. Euchromatin. Chromatin is genetically active part of the chromosome. Genetically active part of the chromosome. And it is stained lightly during interphase, and it remains uncoiled. They remain uncoiled during interphase and they are lightly stained. But when it condenses during metaphase, it condenses, then it becomes deeply stained. Whereas during the interphase, when it remains uncoiled, it is lightly stained. That means the color it takes up is very light because it is uncoiled, it is spread out. But once it condenses during the metaphase, it becomes deeply stained. It is seen as bands. And you can take an example of the bands that we see in the polytene chromosome. They appear as during deeply stained, and they appear as bands. And these bands can be seen in polytene chromosome. Now heterochromatin. This is about euchromatin. Euchromatin is the part that is transcribed. One of the important points here that we need to remember is euchromatin is transcribed into RNA during protein synthesis. So these are the important things that we need to remember. It is lightly stained, and condenses during the metaphase. It is the genetically active phase. That is why it helps in the transcription from DNA to RNA during protein synthesis, and they show a banded pattern. Especially this can be seen in the banded segments of the polytene chromosome. Now we go on with the heterochromatin. Heterochromatin is deeply stained during interphase. They are deeply stained during interphase and they are genetically inactive. Interphase with maximum condensation. Maximum condensation. They are genetically inactive. They are genetically inactive, and they are basically made up of, like I have already talked about, they are made up of repetitive DNA. Repetitive DNA. You can take example AU, AG, AGU, CC, AA. That is repetitive DNA sequence. So, and this part takes some time during replication as well. Replication normally takes place during cell division when the chromosome replicate to give rise to another half of the same type. This is called replication, and they take a very long time as far as replication is concerned. The main region where the heterochromatin lies, is adjacent to the centromere. If this is the chromosome, then heterochromatin is present in this particular region, maximum in this region, and they show banded pattern like this. This is the heterochromatin. These are the heterochromatin, and these are euchromatin. Heterochromatin and euchromatin. Now, heterochromatin can be of two different types: constitutive type and facultative type. In constitutive type, this heterochromatin are always present on the chromosome. They remains as a part of the chromosome. That means this heterochromatin, which is inactive genetic part of the chromosome, it's genetically inactive. This remains with the chromosome always. Alright. Now, this is called the constitutive. That means remains as a part of chromosome. Always. It is always present on the chromosome, in an eukaryotic chromosome. Now the other is, this is number one. The other is facultative. Facultative type. In this case, one of the chromosome becomes inactive. In a homologous pair, one of the chromosome becomes inactive. For example, a female has XX chromosome. Female human has XX chromosome, out of which one of this X becomes inactive. Because its function is not real, because X is already present. So one of it becomes inactive. This is what is also commonly known as bar body. The inactive X chromosome is called the bar body, or it is also called facultative heterochromatin. Constitutive, that means it is composed of, it remains with the chromosome. But facultative, it happens only when the case becomes such that there are two X chromosomes, one of it becomes inactive. So the inactive one is called the bar body. So much for today, boys. We'll carry on further in the next class regarding the Lamprus chromosome and